turned, but what a fantastic turnout for this, what will be, I know, a tremendous inaugural professorial. It's always a real delight to sort of anchor the start of these congregations together, where academics are very, very busy individuals. In this institution, we all absolutely try to live the mission of the organisation, which is transforming lives through innovative and inclusive education and research. And one of the ways that we recognise this is through the attainment of colleagues who've worked for many years, sometimes really leading on education and support for students and the outcomes, and sometimes also really focusing in terms of research with impact. And tonight is a delight because we've got a colleague who focuses on both of those areas and also is able to demonstrate really significant contributions to leadership, not just of his discipline, but within his school and indeed more broadly across the organisation. I've had the privilege of working with Nick for the time that I've been here at ARU, so it is wonderful. It's great to see so many visitors, collaborators and friends and family of Nick here tonight, and I think this is going to be a real treat. It's one of the most challenging things you do in an academic role when you're celebrating your professorship. You're used to talking to colleagues who understand what it is that you do in detail in your discipline. But we're talking to an audience here where we've got family, we've got colleagues from arts, humanities, we've got students, we've got lots of different guests, really very interested to learn more about Nick, and really important also that sort of passion and the topics which really have excited him to help him grow and really demonstrate a fantastic career that he's achieving for himself and that will continue on. So I'm very much looking forward to everything that you're going to say. He's been chomping at the bit because he's just fidgeting walking around. So without any further ado, I'll hand over to Professor Laurie Butler, PVC and Dean of the Faculty of Science and Engineering, to say a few words.
Thank you, Professor Barnett. Thank you very much. Real pleasure to introduce the speaker for tonight's inaugural lecture. So I've also had the pleasure of working with Nick in various guises since I joined ARU. Nick has asked me specifically to keep my opening introductory remarks really brief, which I know is because he's chomping at the bit rather than because he just wants me to keep quiet. But let me just give you a little bit of background for Nick. So Professor Nick Pugh studied biology for his undergraduate degree at Southampton University, graduating in 1996. His PhD was at the National Heart and Lung Institute, Imperial College London, under the supervision of Professor Alan Williams. On completing his PhD in 2003, Nick then moved to Dr. Jeff Butcher's lab at the Brabham Institute in Cambridge. Subsequently, Nick moved to Richard Farndale's laboratory at the Department of Biochemistry, University of Cambridge in 2005, to look at the interaction between platelets and collagen. Now, since starting at ARU in 2013, Nick has been investigating the link between zinc signalling and platelet activation during thrombus formation. This work has been supported by the British Heart Foundation with 2 project grants and a PhD studentship to date. Alongside research, Nick has held significant responsibility in leadership and management whilst at ARU in posts including the Deputy Head of School of Life Sciences, Deputy Dean for Research and Innovation, Director of Research Students, and most recently, Faculty Director of Research and Innovation. He's also a member of the Health, Performance and Wellbeing Leadership Team at the university. Now, in his talk this evening, Nick will explore the central role that metal ions have in biochemical processes and why they are essential for life. So Nick will provide an introduction as to how ions support various biochemical processes, and we'll explore how ion channels are able to transmit ions at incredible speed whilst maintaining near perfect selectivity. He will also provide some of his recent data about how metal ions affect the processes of blood clotting and thrombosis, and how better understanding of ion handling may lead to the identification of new antithrombotic therapies. And he'll do all of that in less than 50 minutes. So, with a talk entitled Metals in Biology, How Metal Ions Contribute to All Aspects of Biology, it gives me really great pleasure to introduce Professor Nick Pugh.
Great, thank you very much. Thank you all for turning up. It's really good to see so many people here, going back a long way. Lots of people from my career, got my family here as well. Just thank you all for coming along. So hopefully you all know who I am now. I'm Nick Pugh. I'm Professor of Molecular Biology. And I think the first thing I needed to do was to work out what it was I was going to talk to you about today. So what happens is you get appointed to a professorship, then they tell you you have to give a talk, but they don't give you any more information than that. It's up to you to decide what it is you want to talk about. So what I did is I put inaugural lecture into Google just to see what it said. An inaugural lecture is a speech given by a newly appointed or promoted professor. That's me at a university. That's here. Yep, we can do that. That's no problem. It's a chance for the professor to share the research and achievements and celebrate their career milestones. Yes, we can do that, no problem. And the last thing is to thank key collaborators, friends, and family who have contributed to their journey. And I think that's what's going to be the most thing here. It's going to be thanking all of the people who've contributed to me and my journey to get to this stage in my career. So I've had a few chats with people, a lot of advice from people who have told me what this lecture should be about and what I should include in it. Some really good comments from my office mates. First of all, make it accessible. So this is not a hardcore science talk. I'm not going to be having loads of graphs and lots of aggregometry traces, even though I really, really want to. It's going to be accessible, so people are going to be able to understand it. My mum's here, for goodness sake, so it has to be a little bit accessible as well, okay? So that's the first thing. We all need to be able to understand what's going on. Make sure it's not too boring. Thank you very much for my colleagues for telling me to do that. There's no pressure on that. And make sure it's entertaining. So entertaining and not boring, I can try and do that. But, I'm an academic in front of an audience. You're now stuck here. I can do basically what I want, so I think this is going to be the best 3 or 4 hours of your life. Don't talk too fast. That's what Helen told me earlier. Apparently I ramble a lot when I get nervous. I talk a lot too fast, so I really need to slow down a bit. So first of all, I'm going to give thanks to the people who are the most important. So firstly, obviously, I'm going to thank the A-team. That's them there. They're all sitting around here somewhere. That's my wife, Shaista. That's my 2 sons, Haroon and Sammy there, and I'm incredibly proud of all of them. I thank them very much for the support. Shaista's obviously supported me for the best part of 25 years. The boys have supported me for less time, but it's not really their fault. But they're always there for me. My eldest son is Haroon there. He's doing biochemistry at Leeds University. I'm incredibly proud of him. When he was young, we indoctrinated him basically into biology. We took him to zoos, museums, science days, to make sure he would become a scientist and a biologist, and we succeeded in indoctrinating him into that. So we're very proud of him for that. And then there's Sammy there as well. Sammy went through the same indoctrination process, and it didn't work. Sadly, Sammy's gone down a different career path. He's into drama, he's into media, he's into ancient history, and I don't know where he's got that from. I do slightly wonder whether they gave us the right baby at the hospital. But he does have a penchant for a ridiculous humour and ridiculous facial hair, so I think he is definitely related to me quite closely. I mean, my boys have supported me. Joking aside, they've supported me incredibly. So for the last 10 years, this has been quite a good 18 months for me. So I got made professor. I got a significant birthday I'm not going to tell you about. And we also, all 3 of us, we got our black belts at karate. So I'm putting that in there because it's quite... I didn't expect that, but I was just going to say, if you don't like the talk, you can have a chat with us afterwards outside. No problem there. Shaista, thank you very much for supporting me. This is a team effort. This is not me. This is us. We have done this as a family. Shaista was a researcher. I met her doing our PhDs, and she changed careers, and now she's a teacher. She teaches at Long Road. But she did take a sabbatical a few years back to do a research assistant post at the Brain Research Centre, and she generated some data, and the annoying thing is she's basically getting more publications from me from this 1 year of work, which I'm finding very galling. Got to thank, my mum and dad. You've got to thank your mum and dad. That's my mum over there. Thank you, Mum, for everything you've done over the years. It's not been an easy job, I know, but thank you very much. And my dad as well. My dad sadly passed away a year or 2 ago, but I think he would have been absolutely made up if he had been here. So thank you to the both of them. I also need to thank the money as well, people who have paid for me. The British Heart Foundation have supported me all the way through my career, from PhD and to grants as well. So thank you very much for paying for my research. The Wellcome Trust gave me a bit of money, but I also want to thank the Cambridge Philosophical Society. They haven't given me money, quite annoyingly recently. They didn't give me any this week, but they have paid for PhD studentships at Anglia Ruskin University that I've been involved in organising, and that's been really, really useful. Really good for us. And of course, I've got to mention this place. I know, what staff are like. We do tend to whinge about working here, don't we? We do tend to complain about senior management and things like that. But if you took a step back and actually think about what we've got here, this is a great university. It's filled with absolutely amazing people, and it gives us the chance as academics to do exactly what we want to do, which is a bit of research and a bit of teaching and progressing well. And I couldn't have done this without having Anglia Ruskin looking after me here, so I just want to extend my appreciation to them. So what are we here to do? We're going to celebrate my career milestones. As I say, this is a talk. It's going to be about me, my ego, and you're stuck here forever. You cannot leave. Oh, that's right. So what we're going to talk about, I'm going to talk about how I got to this point. I'm going to do it very, very quickly. But we're going to talk about different parts of my career. So we're going to have to start at the start of my career. As a younger man, I'm going to talk about my degree. Yeah, I was a looker, wasn't I? Yeah. So that's early years. I'll talk about the early years, my PhD, my degree. I'll do a little bit about that. We'll talk about the middle years when I was a sensible, mature postdoc. This is in Richard's lab. I'm very, very happy about that. I'll talk about that quite a lot. But we're going to have to talk about the future, what I'm going to do in the future, what my future research plans are. And that's how things are going to be, and obviously hair transplants are going to be a major part of what's going on. But I will talk about all of those things in all seriousness. One thing I wanted to make absolutely clear at the moment, this is a research talk. I want to talk about my research, but research is not the biggest part of the job we do here as academics at Anglia Ruskin. We do a lot of teaching, and we do a lot of admin. And I think, that part of my role has also contributed to my journey to be here as well. So as Laurie and Yvonne were pointing out, I was appointed as senior lecturer here, then I became deputy head of department, then deputy head of school, then deputy dean, faculty director of research and innovation. So a lot of what I've done here has been administrative or leadership and teaching, and that's been a major part of my role, and I've really enjoyed doing that as well. And I need to acknowledge that as well. I'm a professor for the leadership, for the other stuff, as well as some of the research and some of the money I've brought in. A couple of the people I want to thank for that. Obviously, when I came here in 2013, Sheila Pankhurst was the head of the department at the time, so I just want to thank Sheila for appointing me. Funnily, she sort of appointed me, and then she decided to leave 6 months later. I'm not too sure whether that was linked or not. I'm pretty sure it was. And also, Laurie down there, thank you very much, Laurie, for appointing me to deputy dean. He only appointed me for 6 months. I think he saw the writings on the wall. But luckily, they couldn't appoint anybody else to take over, so I did the job for a good 3.5 years. So thank you very much. That's been a wonderful experience in senior management. I've really enjoyed it. Is that all? Oh, I've got to thank that other guy, don't I? So that's Chris Paris. Where is he? So I've got to thank Chris up there. Chris has been a wonderful guy for me. So he's been here about 8 years. He's been my friend and mentor. He's looked after me throughout the whole time. I owe him a great deal of gratitude for that. So thank you very much, Chris. Any tear? No? Oh, well, that's what I was trying to get you to do. But these are the people who've helped me here in my teaching journey. But I'm talking about my research. That's what I'm going to talk about for the most part here. And Laurie went through this. This is basically my career here. I did a degree in biology at Southampton University. It was in ecology and animals and environment. That's what I was interested in. But during that degree, I worked out that's not what I wanted to do. I wanted to work in virology and molecular biology and biochemistry. So I made changes and made choices in my modules that brought me down that route. And that's the gang up there that I'm still in contact with. I've got 2 PhDs down there. That's not because I'm a genius and did 2 PhDs. I did 1 PhD and failed at it miserably. And that was at the London School of Hygiene and Tropical Medicine. But things were quite forgiving in those days that I could actually leave that one halfway through with nothing to show for it and start another PhD at Imperial College. It's quite a traumatic story. But if you want to hear about it, we do a podcast, me and Marika. So what we're doing is we're interviewing a lot of biomedical staff here about their careers, and it's to show our students how to get a career in biomedical sciences there. So she interviewed me, and I've got my whole story on there. So please look at that, because I'm hoping it's going to make my fortune as well. Any good YouTube providers, Spotify, it's all on there. Okay. After my PhD, Imperial College London, I basically moved to Cambridge. I was basically following my wife around like a puppy. She got a job up in Babram, so I followed her up here. I did a second postdoc in Cambridge with Richard Farndale, his lab. I'll tell you about that later. And since then, I've been at Anglia Ruskin from then to there. So when I was asked to do this talk, I was asked what was the theme of my career, and that was a really hard job because I've stuttered around in my career from one thing to another, and there's no real solid theme that's gone on through that I can say, this is what I wanted to do, and this is what I'm doing now. I'm inevitably going to talk about these 2 things. These are what I work on at the moment. These are platelets, little blood cells, and zinc, a metal ion, which is sort of the title of the talk. I work on zinc now, and at different times in my career, I've also worked on various metal ions in biology. So I'll give you a little bit of background to this. Metal ions are really, really important. They're essential for life. Here we have a number of different metal ions, such as sodium, potassium, magnesium, all of the way across that list. And there's zinc at the end, which is what I'm particularly interested in. So metal ions are very, very important in biology, and here are just some of the examples. Hopefully you can see up there you've got hemoglobin, the protein that takes oxygen around the body. Hemoglobin has 4 iron atoms in the center of it, and it's the iron atoms that bind the oxygen in your lungs and then transport it across all the way to the tissues to deposit it there so that we can breathe quite nicely. Metal ions are also very important in signaling in things like axons and neurons, where membrane depolarization opens and closes different membrane channels, allowing metals to go inside and out, and that's how we propagate signals down neurons. That thing up there, that blue thing, that is a transcription factor. That's a protein that turns on gene expression by binding to DNA. And that green blob up there, that's a zinc ion. So that's known as a zinc-fingered transcription factor. The zinc ion is really important as a structural part of the protein. It keeps the protein in a certain shape, making sure it can work properly. And metal ions are also important in certain enzymes. Here we've got cytochrome C and carbonic anhydrase. Carbonic anhydrase also has zinc at the catalytic site of the enzyme where it coordinates the reaction. And cytochrome C is very, very important in redox reactions, electron transfer reactions, and it has a copper ion as well. So copper, all sorts of ions are very, very important in our biology. However, my interest has been not in what they do, my interest has really been in how metal ions are involved in signaling. So if you have a cell, what you need to do is you need to communicate with all the cells in your body, so you need signals going around your body all of the time, and metal ions are quite important in this. So calcium is the best known signaling metal ion. So calcium, hopefully you can see my little GIF up there. Many of our cells have stores of calcium, little membrane-bound stores within them, and when you get a signal or a stimulus to the cell, those stores will open, allowing the calcium into the cytoplasm, where they will do things in the cell, depending on what the cell does. Calcium is a particularly important ion. It will drive many different intracellular processes. A lot of the enzymes that are present in cytosol are calcium-binding enzymes, and calcium will basically turn them on. You can see in this video up here, that is a calcium signal. So these cells are loaded with a dye that goes brighter when calcium is present. So you get a signal coming in from the top right. It will activate those cells, and they'll release calcium into the cytosol. You can see a nice flash there, and that's the sort of thing I'm interested in. This sort of calcium signaling happens in many different cell types, such as immune cells, muscle cells, neurons, oocytes as well. During fertilization, the process of fertilization, you'll get a calcium signal generated across the oocyte. And of course, platelets. Platelets is what I'm very interested in. And you see this type of calcium signaling process going on in platelets. So my first introduction to metal ions in biology came in my PhD. This is the second PhD, the one I completed and did well, not the first one I failed miserably at. This is the second one where I did with Alan Williams at the National Heart and Lung Institute. Sadly, Alan passed away a few years ago as well, but he was an absolutely fantastic boss, and I owe him a great deal for supervising me through my PhD. I wasn't a particularly good PhD student, but I made it through there and did some really, really good work in his lab. Now, the model system we were working at in the lab was sort of based on heart muscle cells. This is a cardiac myocyte up here. What you can do is you can get a mouse, you can take the cells out of the mouse heart, put them in a petri dish, you add the right chemicals, then the cell will actually start contracting. And when it's in a heart, basically it's that cell contracting with all the others at the same time that makes the heart beat. Now, what regulates that is a calcium signal. In this cell up here, you can see, once again, this is stained with a dye that picks up calcium. There's a calcium wave that goes across the myocyte, and that's what's regulating that muscle contractile activity. So I've got a little schematic of that here. So what you can see is a muscle cell. On the right-hand side, you can see some calcium outside of the cell there. So what happens, a signal comes to the heart cell, it results in a membrane depolarization, and what that will do, that will open up channels on the membrane, you see in the green one on the right, that will allow a little bit of calcium into the cell. That little bit of calcium coming in will interact with that green blob on the store of calcium, and that green blob is called the ryanodine receptor, which is a specific type of calcium channel. And that's the gatekeeper, that's the gatekeeper to the store of calcium within the cell. So a little bit of calcium binding to the ryanodine receptor will open it, a lot of calcium will come out, and that will interact with the contractile apparatus of the cell that will move the myosin and acting against each other, and that's what causes the heart cell to contract. So this is the biochemical process that we were interested in. We were specifically interested in that ryanodine receptor there, which is that little blob that I just mentioned. Now, the ryanodine receptor is a very difficult protein to work with. It is really, really big, a really huge protein. It's about 65 kilodaltons as a monomer, but you can see here, that's the ryanodine receptor. There are 4 parts to it, 4 corners to it, so it's a tetramer of 4 subunits, and there's a pore, there's a little hole going down in the middle. I wish I could show you that, but my thing isn't working. just in the middle there, that's a hold, and the ions, the calcium ions, will go through that hole from one side to the other down their concentration gradient. It's an interesting protein. We were interested from its biochemical properties. However, it is involved in disease as well. There's a catecholaminergic polymorphic ventricular tachycardia, where you have mutations in this protein that cause heart arrhythmia or difficulties in the heart beating. Now, our interest in this protein is that if we took this protein and we put it in an artificial membrane and we put some salts on either side, we could actually look at ions going through the channel. And what we found out that ions went through the channel faster than we would expect. So they were going faster than the diffusion limit for what we would expect of the solute concentration, which we thought was interesting. So things were going faster through it, but there's no energy here. It's not pumping. So we didn't know what the biochemical properties of the channel were that would allow ions to go through much easier. And that's what we were basically doing. At the same time, another group in the States crystallized another channel, and this is called KCSA. That's called the potassium channel of Staphylococcus aureus, and that's another potassium channel. It's much, much smaller than the ryandium receptor. One of those would fit basically a tiny little bit into the channel there. But this gave us some ideas. We looked at this channel, and it gave us some ideas about the structure of the ryandium receptor, because we saw the same sort of features present in the KCSA channel that we did in the ryandium receptor. So we looked at the DNA and the protein sequence of the ryandium receptor, and we saw the similar sort of things, particularly this structure here. That's known as the selectivity filter of KCSA, and that's an important part of the protein. This is where ions go through the channel, and you have this part of the protein here that is pointing into the middle, and that's forming the pore. Now, the exciting thing about this channel is that it's very, very selective for potassium over sodium. So these are two different metal ions. Now, potassium and sodium are basically two barren, featureless spheres. One is slightly smaller, sodium slightly smaller than potassium. But when you look at the KCSA channel, basically potassium will go through very quickly as though there was no protein there, as though it's just going through a water-filled pore, but sodium couldn't go through it at all. So it is 100% selective pretty much for potassium over sodium, even though potassium was bigger. So if you think about a model of that, if I had a load of cricket balls and table tennis balls, I poured them into a funnel down a tube, what would be happening is that the cricket balls would be bigger, they'll be going all the way through, but the channel would be selecting and not letting any table tennis balls go through at all. And it's quite an odd thing how that could possibly happen, because you wouldn't expect that to happen in a tube of the same type. To explain that, you need to understand what ions are like in water. So that's potassium ion there. In a fluid solution, in a solution, basically potassium becomes associated with water, and these are water molecules on the outside. This is called the hydration shell of potassium. And the distance between the water, the oxygens on the water and the potassium, that's where the binding is a certain distance. It's a hydration distance between the two. And that's constant. So all potassium will have water basically bounding it and surrounding it. So there's the KCSA channel, and what I've done is I've just zoomed into the selectivity filter. That's the bit in the middle of the channel. And what you can see are these red groups on the channel here. Now these are carbonyl oxygens. So these are oxygens on the protein, just like oxygens here in the water. And these are also exerting an electrostatic effect into the center of the channel. So what the channel does, and why I like about it, is it's really quite clever. The distance between the oxygen atoms on the selectivity filter are exactly the same distance as the oxygens would be in the hydration shell of potassium. So for the potassium ion to get through the channel, all it needs to do is substitute the oxygens in the water for the oxygens in the selectivity filter, and it can just slip through the channel as though it's just going through water. So if you had eyes and you were a potassium ion, you would just see oxygens around you, whether you were in solution or whether you were going in the channel, and that's why it goes so fast. But then if you think about sodium, sodium's slightly smaller, and it has a different hydration shell around it. It's got different properties. It's not quite the same size as potassium. So this means that you cannot dehydrate the sodium very well by the selectivity filter because it's not exactly the right size. So a sodium ion will come along, it'll try and get through, but it cannot dehydrate, and it will go wandering out again. So that is why potassium could go through the channel really, really well, as though it's not there, and that is why sodium cannot. So these were inspiring us, these questions, at the time of my PhD. But the question I wanted to ask about the ryandium receptor was why were the ions going in much faster than should have been their diffusion limit? So what we did is we took the KCSA channel, the structure of it, and we mapped it onto the ryandium receptor to see where the amino acids would be if the ryandium receptor had the same sort of structure as KCSA. And what we noticed, there were a lot of negative charges, lots of negative charged amino acid residues in the mouth of the channel. And what we hypothesized is that maybe all these negative channels are going to attract a lot of positively charged metal ions, increasing the concentration of these at that level so that they can go through the channel more quickly. And that was the hypothesis we were working at. So what I was tasked with doing in my PhD was to take the ryandium receptor and to mutate those amino acid residues from a negatively charged to a neutral charge to remove that effect to see what would happen, see whether they would go through. So I'd love to talk a lot about my PhD, but this was a very, very big task. This was 3 years of me cloning and mutating the ryanodine receptor. I didn't really get round to the last stage of actually doing the proper experiments to see what it did. The problem was the receptor, the protein, would spontaneously mutate during the cloning procedure, and it was very, very difficult to work with. But I spent 3.5 years trying to clone it, trying to express it. I succeeded in the end, and I managed to look at it using some single-channel equipment. Wasn't a particularly successful PhD, but what I did manage to do was to publish some of my control data in a paper that I have up on the board up there. So it wasn't completely useless. I did get a paper out of my PhD. And the best thing about that paper was that as part of it, what we needed to do was to work out the protein sequence of the sheep ryanodine receptor, which we didn't know at the time. So this is in my next job. Basically, I took some sheep heart, because I was at a working farm. A sheep heart, took out the messenger RNA, converted it into DNA, and sequenced it. And I found out the sequence was pretty much exactly the same as the human, the rabbit, the sheep, and everything else. But it was an important experiment to do because we needed to be able to model what we were doing. So that was my PhD. I got my PhD, I think, in 2003-ish. And after that, I followed my wife Shyster to the Bagram Institute. I got a postdoc in the lab of Jeff Butcher, who was at the Bagram Institute at the time. I was only there for 2.5 years. It wasn't a particularly successful time as well. We didn't get many publications at all after that. But I was doing the same sort of thing. I was cloning and expressing and mutating proteins. In this case, it was a protein called GuyMap, or a small GTPase. So I won't talk about that too much, but I do want to talk about the time I spent in Richard Farndale's lab. Richard's up there. Thank you very much for coming, Richard. Once again, you employed me for 8 years, which I still think was a mistake, but you did it anyway, and I'm still very grateful for everything you did. And I had a wonderful time over those 8 years in Richard's lab. That's Joe up there. Joe's up there. We had a few beers a few times. And Richard's a very, very serious, sensible academic, so that isn't a photo of him taking a picture of me being seasick and hungover on a boat, because he would never do something like that. But I had a great time in Richard's lab, and we did a lot of good science, I think. This is where I first became introduced to platelets. Now, platelets, I need to explain a little bit about this. These are blood cells. These guys know about them, don't you, because you've been to my lectures on them. So platelets are blood cells, and what they've got, a single job is to detect areas in your circulation where you've damaged yourself, where you've cut your finger or where whether you've damaged your circulation. So if you look in this image here, these are platelets here. This is a blood smear. You've got some red blood cells, white blood cells, and platelets in there. Those are the little platelets. They're incredibly important cells because there are certain diseases where people don't have any platelets at all, called thrombocytopenias. And in thrombocytopenia, you get this sort of these symptoms here, where you get these little bruises formed under your skin. Now, there's no damage there, so what's happening is your veins, your arteries, they're spontaneously breaking, and blood is leaking out underneath the skin. And that's telling us the platelets are working all of the time, just to maintain our circulation. They're maintaining a healthy circulation all the time. So they work very well to prevent us from bleeding out when we cut ourselves, and they're working all the time to repair our circulation. There's a dark side to platelets as well, in that they're the cells that are responsible for heart attacks and strokes. So in that image here, this is a coronary artery, I think, and what's happened here is a fatty deposit on the coronary artery is broken. It's released thrombogenic material into the circulation. Platelets are bound to it, and they've formed a clot. Just like they would do, for example, if you cut your finger, if you cut your knee, you've got a clot formed. The only difficulty here is that it's in the wrong place. It's in the middle of the blood vessel. It's stopped blood flowing through the vessel, and it's caused ischemia, or prevention of blood flowing to a tissue such as the heart, and that causes a heart attack. So a lot of work I'm doing is based on the fact that we are studying cardiovascular disease, and I think you should understand that later. So what do platelets actually do in the body? So that's a platelet up there. What they normally do, they circulate around. They're basically looking for sites of damage. And as soon as they see a site of damage, they will interact with it, and they will help to repair it. To do that, what you need to do is you need to take your platelet, which is senescent, not doing anything, and you need to stimulate it a bit. And when you stimulate it, a number of things happen, and there are 3 of the important things on the board happening there. So the first thing is you get a shape change. Your discoid platelet will start extruding these filopodia, these little tentacle-like things, and they'll spread out into this large fried egg structure. And what it's doing there is it's spreading itself over as big an area as it possibly can to prevent blood loss. The next thing platelets will do, they'll stick together. They'll aggregate to form a platelet clot, a platelet thrombus. And that's shown in that video there. They get very sticky, they stick together, and then preventing forming a plug through which blood can't go. And the next thing is they will release their granules. That's an electron micrograph of a platelet up there. Platelets have got a lot of granules in them with a lot of bioactive material, such as coagulation factors and growth factors as well. So when a platelet becomes activated, it releases the granules, it releases growth factors and things, and helps to repair the damaged area. Granules also contain zinc, which I think is something I want you to remember for a bit later on. So you need stimulation for a platelet to actually do something. So we have to ask ourselves, what is it that's stimulating the platelet in the circulation? So I've got on the board there, that is the structure of a blood vessel wall. That's an artery wall. You have these endothelial cells up here. Hope you can see my mouse up there. Endothelial cells, and they prevent blood from getting outside of the vessel. That's the first line of defense. Now, immediately under the endothelium, you have this extracellular matrix layer, and that contains a lot of the protein called collagen. Now, we all know what collagen is, hopefully. It's the sort of thing supermodels inject into their buttocks and their lips and things like that. I wouldn't recommend it. But it is an incredibly important protein for our body. It's what our cells stick to, is what essentially keeps us together. So I've got this little GIF here. This is basically a side-on view of a blood vessel. You have your endothelial cells. Underneath that is a layer of collagen. So if you damage your endothelial cells, the first thing the blood is going to make contact with is this extracellular matrix of collagen. And the important thing to notice here, collagen is what platelets interact with. Platelets bind to and identify collagen, and that's what we were investigating in Richard's lab. So I've shown on here on the left hand, this is a video I took in the lab. So what I've done is I've got a microscope, I've got a glass slide, I've coated it with collagen, and I've perfused blood across that slide. The platelets here are labeled in green, and you can see the platelets perfusing from right to left. They will bind to the collagen, they'll become activated, they'll start aggregating together into these little aggregate lumps, and that's what I was doing essentially in the lab. For those of you who like platelets, on the right-hand side, I've got some light transmission aggregometry figures. You don't need to worry about these too much, but basically if you add collagen to a suspension of platelets, they will start sticking together, coming out of suspension, and that's what's shown on that figure down there. So platelets and the collagen interaction is very, very important in our hemostatic reactions that keep our blood within our body. So what's happening in a very basic way is shown in this little video here. So what's happening is that little endothelial cell here, that's going to fall off, that's going to die, and expose some of the collagen to the flowing blood. So that's what happens there. And what happens is the platelets, which are the little brown cells, they will bind to the collagen, transiently at first, and then firmly bind. They'll undergo a shape change, so they'll spread, they'll start ejecting some of their granule contents to start releasing some of the chemokines and cytokines that will encourage repair and growth of that region. And that plug that they form, that will prevent any blood loss from that different region. And this is the sort of thing that happens in our bodies all the time. So when I was in Richard's lab, the question we were asking is, how do platelets interact with collagen? What's the way they do it? And what is the result of them interacting with collagen? So in Richard's lab, I don't want to be too unfair, it's not a platelet lab, it was a collagen structure and function lab. So their view, when I started there, they were looking at the collagen, what it does, and the structure and the function of it, whereas platelets were the best cell model for how biological things interact with collagen. Now Richard, he had this great strategy for actually looking at collagen and working with it. What he would do, he'd take the structure of collagen, and then he would get a peptide synthesizer, and he would synthesize artificial sequences of collagen that would then spontaneously form into their normal structure as they would do in the body. So he was able to generate some small collagen peptides to which we could start looking at how platelets and how other cells and how other proteins interact with collagen. So if you take in this example here, well, I've got three collagen interacting proteins, and we all know that these would bind to collagen somehow. But what Richard did, he basically took that collagen molecule and he synthesized many, many smaller overlapping fragments. And this was really, really useful, because now what we can do, we can identify exactly which sequence of collagen that protein would specifically bind to. We can then isolate it. Obviously it's synthetic, we can do what we want. Isolate it away from all of the other collagen, then we can look at single specific protein ligand interactions, which is actually quite difficult to do. So by doing this, we could identify the collagen binding site, and we had a ligand to control our experiments. So platelets have 3 collagen receptors on their surface, and they're shown in this little video here. You have glycoprotein 1b59, glycoprotein 6, and integrin alpha-2 beta-1. We knew they were there. We knew they were collagen-binding proteins, but we don't know what contribution these receptors made really to platelet activation and to what platelets did. So what Richard was able to do and what we did in the lab, we could basically take our collagen substrate on the bottom, we could get rid of that, and we could replace it with the peptides that he made, the receptor-specific peptides, either one individually or in combination with other ones. So we can basically start to tear apart or to really investigate what the different collagen receptors on platelets were doing during platelet activation. So those are the peptides there. He's got CRP, collagen-related peptide, GFO-GR, and VWF3. And what my job was to do was to take these three peptides that bound to the 3 collagen receptors and look to see how platelets would interact via their collagen receptors to them. So my job was to basically get on this, this microscope here. This is a confocal microscope. It had a flow chamber on top of it through which we perfuse blood to make contact with these peptides, and we would look at different types of platelet behavior. And these are the sort of videos we see. The platelets here are labeled in a fluorescent dye. If we engaged all 3 collagen receptors at the same time, we saw what we saw earlier, which is basically platelets binding to the collagen, activating, forming a thrombus. This is the sort of thing that happens in your body all the time. So these are the platelets behaving really quite normally. If we only used one peptide that bound to the receptor glycoprotein 1b59, we saw something different. So what's happening here is these platelets, they're not becoming stable, they're not sticking down. What they're doing, they're rolling across the surface of the collagen, not becoming stuck. But there is a transient interaction. They're binding, they're slowing down on it, but they're not stopping abruptly. If we engage the 2 other collagen receptors here, that's glycoprotein 6 and integrin alpha-2 beta-1, you saw another different type of behavior. Here, what's happening here is you're only getting 1, 2, 3, 4, 5, 6 platelets binding firmly to the collagen. So they can't bind very well to this surface, but once they do bind, they become fully activated, and they're able to recruit other platelets to form these quite large clots at the end of the experiment here. You see that? So what we're able to do is to use these peptides to dissect out the behavior of these cells. I won't go into too much detail about what we found here, but this was a blood paper, which is probably the best type of paper I could have published in at the time. This is the work we published in there with us and our collaborators there. And what we found is that glycoprotein 1b59, that was the initial adhesive step for a platelet. It would slow platelets down on collagen, and that would allow other receptors such as glycoprotein 6 and integrin alpha-2 beta-1 to then mediate firm adhesion of the platelet to the collagen and then regulate platelet activation. So you get activation of other receptors such as this integrin up here, integrin alpha-2 beta-3, and that was the model we were working at. During this time as well, I started getting interested in calcium signaling in platelets, as I mentioned earlier. So these are platelets, they're not loaded with a specific dye, they're loaded with a dye that responds to calcium. So these are platelets rolling across the surface, and as soon as they become activated, there will be an increase in intracellular calcium within the cell that makes these cells go really nice and green. And this is a technique that I was doing quite a lot in the lab. And what I found is I could isolate different types of signal in the different cells depending on what receptor they were binding through. So if they had the GP6 receptor, you would get a really nice spark of calcium at the start, and that would be followed a few seconds later by a really long sustained calcium signal. Whereas if you just had integrin alpha-2 beta-1 or glycoprotein 1b59, you would only get calcium sparks. You wouldn't see that sustained signal. So that's what I was doing in Richard's lab. Once again, very grateful for all the time I was in there. But 2013, that's when basically I was looking for jobs. Well, I wasn't really looking for jobs. I thought, I'll apply for this one for the experience, and accidentally got it. And that's genuinely what it felt like over there. I felt I was absolutely drowning in becoming a senior lecturer when I was happy being a postdoc. I didn't see why I had to get a proper job, but I just apparently had to. Because I came there, I couldn't keep working on the same stuff that I was doing in Richard's lab. I've been 8 years in there. I was basically indoctrinated in the lab. I had to find a new idea, so I had to start thinking about what else I could possibly do. And that's where I came across zinc. So Samir's up in the lab up there sometimes. So Samir, he was doing a lot of recombinant molecular biology in the lab, and he was purifying proteins, different type of proteins, on columns, and eluting the proteins using zinc, because it bound to the nickel. I think these proteins were not stable at the time, so he was adding zinc to them to stabilize the polyhistidine tail. Don't worry about that too much. And I thought at the time, well, you can't do that, because if you're going to put these proteins on platelets, then the zinc is going to have an effect on the platelet. I don't know where I got that idea, but it made me do a lot more reading of the literature and got me thinking a lot more about what platelets and zinc and how they interact. I knew that there was a relationship between platelets and zinc, but at the time I didn't really know what it was. But I did a bit more reading about zinc, and zinc is actually a really, really important cation in our bodies. We don't store it anywhere, so we need to eat meat predominantly to get zinc inside our bodies, and we use that zinc in a variety of different biological systems. It's very, very important for growth and development, and dietary zinc deficiency is widespread in the world. It's thought that about 25% of the world's population are at risk of dietary zinc deficiency, and this is particularly bad in Pakistan, where it's seen in 20% of the children growing up in Pakistan, particularly rural Pakistan. Globally, it's been estimated to be responsible for 1.4% of all deaths, and obviously it's not causing all of those deaths, but it's going to be a contributing factor to deaths for other reasons. So I got interested in zinc and what it does in biology. It's not a well-studied area. Calcium signaling is really well studied, but zinc is very, very much less well studied. There's a small group of weird scientists who are just dedicated to it, but most of the field is interested in calcium signaling. That's because there's a lot more calcium signaling, a lot more calcium in cells than there is zinc. But I did find out from the reading that zinc has similar roles to calcium. It is a secondary messenger in some cells. So it changes in concentration in cells following external stimulation. And this happens in a number of different cell types, such as cardiomyocytes that I was talking about earlier, heart cells. Also glutamatergic synapses, shown in this little video up here. When you're getting release of your neurons across your glutamatergic synapse, that will also have a lot of zinc in those vesicles, and that zinc affects the downstream signaling process of your synapse. So as I said earlier, calcium goes from a nanomolar to micromolar concentration. Zinc goes from picomolar to nanomolar, so it's very, very difficult to see. And I think it's probably been underestimated for its role in different biological systems. People had worked on zinc and its effect on platelets previously to me having a think about this. This Boyd Odell, once again, he passed away quite recently, very sadly, but he did a lot of work on rodents and humans with dietary zinc deficiency. He found that if you took a rat and you gave it a diet without any zinc in it for a few weeks, and then you saw what its hemostasis was like, you saw whether it was bleeding okay, you could do a little tail wounding assay and you found it had a prolonged bleeding. So these mice would bleed for a lot longer than they should do, just because they didn't have zinc in their diet. And what he did, he took the platelets out of the animal and he saw that the deficiency was with the platelets, it wasn't with anything else. Similarly, in humans as well, you can actually induce experimental zinc deficiency. You just get some humans, you don't give them any zinc in their diet, and you'll see the same sort of thing. You take their platelets out, their platelets are not as functional. But then if you give them zinc supplementation in their diet, that will resolve itself. So zinc seemed to have an effect on platelets that I think wasn't well studied at the time, and I thought, well, here, this is great. This is the gap in the knowledge. I need to find out, I can develop an idea around this. So I had an idea, but the next thing I needed to do was to get research funding. Now, I mean, many of the academics here will know how difficult it is, and I tried a number of different routes of getting research funding. This was the most successful one, I think. In the end, after a couple of attempts of, literally begging, I got some funding. I got a project grant from the British Heart Foundation, which was really, really welcome. Thank you to the British Heart Foundation for supporting me there. And that's Steve Abbott. He worked with me very hard in those early years, and Steve's here today, to help me get that grant. I'm very, very appreciative of all that. So we did some experiments in the lab. We took our platelets and we took some zinc, which is very cheap. You can buy it from any old shop. And we sort of repeated experiments that had been done in the past, that if you took some platelets and you put some zinc on them, they will activate the platelets. So there's our zinc. We put that on, that will activate the platelets, and they will form together. They will aggregate together to form a clot. And I thought, well, this is very, very interesting. This is an area of hemostasis that's not really well studied at all. For those of you who love aggregometry traces, I think there's 1 or 2 or 3 of us in there, that's basically what we showed. And what you'd see from those traces, you need a lot of zinc, really quite high concentrations to get a full effect. And that's always been a bit of a worry of me. Is this a real physiological thing, or are you just chucking too much stuff on? We tried this with other cations as well. So there's zinc going that has an effect. Copper seems to have a mild effect. I've always wanted to wonder what that was, but I've never got around to it. But no other cation we tried had a similar effect to zinc on platelets. So we thought this is biologically quite interesting. It's certainly worth being investigated further. We did some more experiments, some more biochemical ones. This is the most important one here, that we've inhibited the platelets, the way the platelets stick together, so they can't stick together now. And we then treat them with zinc, and we see no aggregation. So we're saying from here that this is real biological response. So this is the platelets not behaving well. We're not bursting them. We're not killing them. They're just generally not working. That basically zinc is activating the platelets. And we saw other things like tyrosine phosphorylation, which is a signaling mechanism that occurs in cells when they're being stimulated. So we were quite confident that this is a real effect. This is not something weird. But we are using a lot of zinc to get the full effect. And the sort of zinc that you probably wouldn't ever see at any part of your body, but we can maybe discuss that a bit later on. So we did some experiments. We thought, well, if we took a very small amount of activator of a platelet, here what we're doing is looking at very low levels of collagen here. That's essentially collagen, that's CRP from Richard's lab. If you put a small amount on, it doesn't do anything with platelets, but if you put a little bit more on, then they start sticking together. So 0.3, they will stick together a little bit like that, and that's fine. But then what we thought we'd do is, well, why don't we give the platelets a tiny bit of zinc, so a concentration of zinc that you could possibly see as occurring in a physiological, in the body. And if you see that, what's happening is the platelets get more responsive with a low concentration of zinc. So you're giving them basically a 2-hit model, a small amount of zinc, the sort of thing that you can get from the body, and also another agonist, then you're getting much, much better platelet activation, and the platelets will all aggregate here. So basically a two-signaling approach would work to activate the platelet, and I think that's a much more sensible thing to happen in the body. Rather than having one signaling system doing everything, you need multiple ones to control the reaction. So where does zinc come from in the body? Well, zinc is present in your subendothelium, so underneath your cells there's your extracellular matrix, there is zinc in there, so if you damage that, then some zinc is released. You also get zinc in your atherosclerotic plaques, so if you're like me, you've been eating too many burgers and things like that, you've got these atherosclerotic plaques built up on your arteries and veins, there is zinc in there, so if those rupture, that could be another site of zinc generation. There's also zinc in the platelets themselves. These are some of the granules in platelets. There's a dense granule and an alpha granule, and there have been reports showing that zinc is present in these alpha granules, and these granules are released upon platelet activation. So when you're getting a thrombus formed, these are releasing the granules and increasing the concentration of granules around where the platelet is. And that's shown here. That's an activated platelet. It releases its zinc in the local environment, and it's possible that other nearby platelets will then respond to that zinc to get more activated, so you're getting a nice thrombus built up that way. So zinc, not a widely acknowledged physiological agonist, but there are ways it could potentially be having an effect here. This is a video I took a long time ago. Don't think about this too much, certainly not you, Matthew. But what's happening here is this is a platelet labeled with a zinc-specific dye, and after some conditions that are not physiologically relevant at all, what you can see is over and over again, this zinc is pumping out a little flash of zinc. So it looks like the platelet is releasing zinc into its local environment. And that's what Emily up there, hello Emily, she's working on that. She's my PhD student at the moment. She's looking at zinc release from platelets, quantifying it, how much is released, where it is in which different model systems, and this is some of our early data here. I've said sadly not statistically significant, but if you activate platelets, you do get an increase in zinc in their local environment. She's repeating this. And this is the work I'm doing with Mike Watkinson from Keele University. He's provided us with some specific dyes that are membrane-bound dyes that will help to identify where zinc is coming out of a platelet. So I want to thank Stu Sage as well. I'm not too sure if Stu is here, but when I first started at Anglia Ruskin, we didn't have the right equipment, so we went over to his lab at Cambridge to do some experiments. This is one of the experiments we did. We basically put high concentrations of zinc onto our platelets with a zinc sensing dye in, and you'll see an increase in signal, which we took to think the zinc is getting into the platelet from the outside. And you can sort of see that in this video here, which once again isn't a great video, but it shows the platelet getting brighter as more zinc is getting into it. So we thought this is maybe one of the ways that zinc is doing something, is getting into the platelet and having an effect. These are two of my postdocs who've been in the lab. That's Kirk Taylor and Sahithi Karavi. They did a lot of work on this. So Kirk in particular, he was my first postdoc here. He did some experiments to try and identify how the zinc was getting into the platelet using a number of different channel blockers. Sahithi followed this work up a bit later on. And what we found out, two particular types of channels, trip channels and the sodium-calcium exchanger. If we block those, then we would block zinc-induced platelet activation, which we thought was quite important. So I'm coming to the end of that bit of work. So this is my conclusions here, that zinc is released by platelets in a growing thrombus, and it could potentially have an effect on the behavior of platelets in that thrombus. And the zinc will contribute to platelet activation, whether it's the one thing that does anything, or whether it contributes in part in addition to other agonists, we don't know. However, what we could say is we could possibly target zinc-induced platelet activation as a new antithrombotic strategy for those individuals at risk of heart attacks and strokes, because we are talking about cardiovascular disease here. So during this time, I had a PhD student called Niaz Ahmed. He was looking at various aspects of zinc signaling in platelets, and what he observed was something that was quite interesting. So he would stimulate platelets with a zinc-sensitive dye in, and there was no zinc on the outside at all, but he would see an increase in the fluorescent signal, which suggests that there was an increase in zinc inside the platelet, irrespective of the zinc on the outside. So it's almost as though there was a store of zinc inside the platelet that would be released in a similar way to what you would expect for calcium signaling, and those are some of the experiments he did there, which made us very interesting. This is a novel finding. An intracellular store of zinc that can be accessed following platelet activation was quite interesting, and something we're still working on. The big question we had is, what is the zinc actually doing inside the platelet? So hopefully you'll remember there are three things that platelets do. They undergo shape change, so they change shape to this sort of fried egg shape here. They'll aggregate, they'll stick together, and they'll release all of those granules. So those are the three things that we think platelets do when you activate them, for example, with collagen. What we did is we found some chemicals that would artificially increase the intracellular concentration of zinc. These chemicals called cleoquinol and pyrithione, they're called ionophores. And what we did is we used these to increase the concentration of zinc within the platelet to see what effect it has. And these are some of the experiments that Niaz did. So in this graph here, what you can see is this is looking at activation of platelets becoming activated and basically aggregating and sticking together. If you add some of our zinc-increasing drugs, then you get an increase in platelet aggregation. So the platelets will stick together. And so that's the one thing here, platelet zinc, increasing in platelet zinc inside the platelet seems to result in platelet aggregation. Similarly, we did other experiments here. There's icoconol and pyrithione. If we added that to the platelets, then we would see granule release. It looked like if you increase the zinc concentration, that would encourage granules to be released from the platelet. So the two things I mentioned, aggregation and granule release, it seems that zinc is having an effect there. We also looked a lot at the platelet shape change. Do platelets change shape in response to zinc? And the answer seemed to be yes. So in these experiments here, what we can do is put some of these chemicals on the plates, and we see a downward deflection in aggregometry traces, which we thought is indicating a change of shape in the platelet. If you do experiments to remove all of the zinc from the platelets, then you can completely abolish platelet shape change as well. We did other experiments as well where we looked at the phosphorylation of myosin light chain. So other things that were involved in shape change, those are also changed in the following incubation with our calcium ionophores. And we did further control experiments just to show that this is a real biological response and it's not something that's happening a bit haphazardly and a bit odd. So this paper was published a few years ago, Niaz as the first author, and it basically shows us the same sort of things, that platelets are involved in aggregation, shape change, and granule release, the major things in platelets. I don't know if Maria's here, I didn't see her earlier, but this is Maria, who was another postdoc in our lab. She went down a different route in our lab. She was looking at the generation of reactive oxygen species in platelets, and she found that if she incubated our platelets with zinc, you would get an increase in reactive oxygen species. Once again, she did a lot of work on the signaling interactions there that I'm probably not going to go into very much here. But did a lot of good work, and we published that a couple of years ago as well. It just seems that the reactive oxygen species is downstream of zinc, and that activates other platelet signaling pathways. So this is the big picture. I don't think I've gone on too long. This is collagen on the left-hand side. It's activating a platelet, and it's causing shape change, aggregation, and granule release of the platelet. And what our experiments seem to show is if we did the same thing just with zinc, you'd get the same sort of effects. You'd still get an aggregation response. You'd still get shape change, and you'd still get granule release. So it's looking like zinc is acting like a platelet agonist. And that's essentially where I am now. So we've put together this figure that I want you to learn and memorize on your way out. These are the signaling pathways that we're working on. These are the hypotheses that we're working on at the moment. If you want to go in there, you can look at zinc being released from platelets or from atherosclerotic plaques, getting into the platelet and causing things like granule release, shape change, phosphatidylserine exposure, which I've not spoken about, and ROS generation as well. Lots of interesting things going there. But that's where I am at the moment. I've got Emily working on that as well, and we've also got some other ideas at the moment. So I need to think about the future, what I'm going to do going forward. So like I say, Emily is looking at zinc degranulation in platelets, so zinc coming out of platelets, whether we can quantify that and look at that. I also have Darissa. Where's Darissa? There she is. She's very shy, so I'm going to point her out to everybody. Stand up, Darissa. No, you don't need to. Don't worry, don't worry. Darissa, she's doing a different project. Now, platelets don't have a nucleus, so we can't genetically manipulate them like you can do with endothelial cells or other cells. So what she's doing is trying to develop new protein tools, new recombinant tools that will get into the platelet to change a signaling pathway. And we're looking at that as a tool to investigate platelets and what they do, but there is obviously a potential drug-based clinical approach to this as well. So I'm planning on doing more platelet zinc research. As soon as I can get more stuff done in the lab, I think we've got a lot of preliminary data and a lot of interesting things to do as well. I am getting involved and interested in thrombocytopenia. I saw Raina coming in as well, and I saw Mike and Claudia. There's Claudia up there. We're working together on projects involving looking at antibodies against against platelet proteins that you would see in thrombocytopenia. And I've also done some pedagogic research as well. I've got some data that I'm working with at the moment. So that's basically all I want to say about my research. I do need to say final thanks to everybody who's basically worked in my lab. There's Niaz, there's Kirk, those are two of the early people, and Janae was one of my early PhD students I helped supervise there. There's Maria and Sahiti as well, two later postdocs in the lab. Then we've got Plamina. I saw Plamina. Hello. There's Plamina up there. There's Darissa again. Can you stand up again, Darissa? No, you're all right, don't worry. Emily, and we've got Claudia. I saw Claudia at the back there. Who is working at Cambridge Protein Arrays but did a PhD with us as well. So thank you to all the guys who've worked and contributed to my data in the lab. I also want to thank my collaborators as well. Now this may come as a surprise. Some of you guys might not know you're my collaborators, but you are, and there's no way you can get out of it now. So Matthew Harper, it's a long time. Thank you, Matthew, for everything you do. If I've run out of ideas or I'm a bit down, I'll go to the pub and he'll tell me what to do. He's that bright. He knows my science better than I do, so he's a real useful resource for everybody in the place. So thank you very much for everything you do there. You've got other people there. You've got Amanda, you've got John, Joe's up there as well. Hi, Joe. Other people I've collaborated with, you've got Mike Tausick down there, Mike Richardson, Raina, Stu Sage, and you've got Cleona as well, Cleona Dungleg from our faculty, and Marty Chambers, who we're working on this pedagogic program as well, and also Safe as well from another company I'm not allowed to tell you about, but I'm working closely with him. I want to thank some of my mates as well, some of my collaborators as well. I'm going to ask you to stand up just to show what you look like. Javi as well, where's Javi? There you are, hi Javi. Javi, she's so professional, she's so nice and lovely. I whinge at her all the time, and I can see this look that she's sick to death of me. But she's so polite, she doesn't say anything, she just lets me do it all the time. I whinge about her all the time. And seriously, we work together, we're co-supervising Emily. Thank you very much for everything you've done for me as well. There's Clet, I don't know where I got that photo from, but he's wearing an I Love Nick Pugh badge. I don't know if you can see that. Clet's great in the lab. I appreciate everything you're doing, certainly with the work you're doing. Clet's great, he gives me ideas. He's a wonderful person to work with. That cartoon is Marika. I haven't got a photo of Marika, but you can look at her, she's over there. There she is. Marika, we're working together. Three of us, we want to work together. We want to do some good science in the lab. These guys are really helping me. They're helping me with Darissa. They're supervising her in the lab, and I appreciate that very much. Carrie's over there somewhere. Thank you. We supervise Plamina together. Plamina, where's she? There she is. Yeah, congratulations, Plamina. I just said hello to you. Yeah. But Plamina got her doctorate recently, so very happy about that and the collaboration we've got there. I want to mention some other people as well. So Angela is our technician in biomedical. I've got her there because I've known Angela since I got here the first days. We worked very closely. She's absolutely lovely, and she's their representative of all the technical support we have here in the university, but I want to thank her. Paul Trevelyan, I'm basically on the phone to him. When I was deputy, I was on the phone to him pretty much every day, talking about money, how we could siphon it off, how we could get some of it ourselves. Never worked, but he was really, really good. And Laurie Glendin, Laurie Chatterjee, I know she was helping with my grant as well. Very, very appreciative of her. I've not seen them here today, but the research team, I'm on the phone to them pretty much every day at the moment. There's Charlotte, Sam, and Nikki. They are wonderful people. Sam, I think I talk to him more than I do my wife, but there's nothing else in it apart from that. And Amy represents the Rido Bid management team. I used to speak to Amy on a daily basis to talk about the grants and the things that we were doing. Once again, she's a wonderful person, very supportive, and her whole team there, Steve, Rosie, and Ellen. I want to thank them for everything there. I want to thank everyone else at ARU. It took me ages to put that picture together. That's everybody who's got a photo I could find from the faculty and a little bit elsewhere. So hopefully you can all see your faces on there. And I'll particularly mention the other guys who became professors at the same time as me. Dan, I saw Dan somewhere. He's there. I'm going to have to come to your one now, aren't I? And everybody else on the list there, I'm looking forward to your plenary lectures. Hopefully they're going to come up soon. But I just want to finish with a final thought. My work is about cardiovascular research. It's about preventing heart attacks, preventing strokes, and things like that. So a lot of money has gone into research in cardiovascular disease, and we need to think about whether that is a worthwhile thing to do. There are lots of drugs, but we do have aspirin. Aspirin works very, very well. But we know what causes heart disease. We know what causes cardiovascular disease and problems, and it's this sort of thing that I obviously have too much of. If you do that, if you eat too much unhealthy food, then you're going to find yourself in a bad way. And what we really need to do as a moral story is to think about how we can change our lifestyles, how we can be better, how we can eat the right foods, and if you do that, then sooner or later you'll have a body like mine. Benny, thanks everybody. Thank you all for coming. That's all I want to say now. I'm happy to answer any questions you've got. But just thank you very much for coming along. I hope you had an interesting talk. Is Steph here? Where's Steph? There she is. Steph did this picture of me. This is an AI-generated picture. She put in, man with no hair, with lots of blood, who likes pictures of space. And I've come out really ripped.
Moving on. That was a wonderful talk, Nick. So you set out to be accessible, some fantastic animations. You were really honest and truthful, and I think as a person, to start a talk like this with thanks and to finish it with thanks says so much about you. Just such a terrific colleague. It was stuffed full of science, and it's really great the journey that you took us on. You were really honest when things didn't quite work out, but once you described whenever you found your niche, you're in your niche, you've got plans for the future. You talk about team science and lots of people that you acknowledge, because these things you never do when you're successful, you never do on your own, and I think that's really, really terrific. So I think this has been an outstanding talk. but with his generosity, he's agreed to answer some questions. We probably will have a roving mic, so and anyone online that wants to ask a question, if we can just pick those up, and we'll do our best to answer those. So I think we've got...
oh, not you. So we've got questions. Not you either.
There'll be a tricky question
and
from
you could
Chris
So what I want to,
Morris.
there's, not everybody wants to listen to the question, but over lockdown I started a new hobby, which is astrophotography. So I'm going to show you some of my pictures so you can look at those if you want. Talk about those. Those are much more fun than what I'm going to be doing. Jane made me do this. I wasn't really going to do this, but go on then, Chris.
Thank you, Nick. That was brilliant. I really enjoyed it. You talked in a part of your talk about how platelets and zinc mediate an interaction between platelets and the collagen, the extracellular matrix. Is there an angle on this in terms of platelets interacting with the endothelial cells? Because if you punch a big hole in the side of a vessel, you've got to start your endothelial cells dividing. And one wonders if there's an angle or if there's anybody looking at that aspect. And for me, because then that gets into cancer, into anti-angiogenic therapies. So is there anything known around zinc and endothelial cell
That's an
proliferation?
excellent question, and Javi's going to answer it because she's 2 seats to your right. No, I think there is. We're talking. I mean, it's part of Emily's PhD to look at the zinc interaction with endothelial cells and the subsequent platelet interaction after that. I can't exactly remember what we were doing, but I was in my own office and they were talking about zinc sulfate today in their wheeled endothelial system. So yes, that's true. I don't know exactly what to do about that, but it's something that we have thought about and something we do want to follow. You know, I'm very lucky. I've got people like Javi here. Once again, she has to cope and put up with me, but you know, she knew that's part of the job. But she's got people here who can work with me on that sort of thing, so it's a great place to be.
I think we've got another question here.
It's a nice one.
Are you sure?
Do you ever get extracellular zinc in the body? Or does it ever
That's a
go
good
up
question.
under,
Thank you.
under pathology, and could it affect clotting?
Do you get it? Well, not in any way that's sort of been proved or demonstrated, and that's one of the things we want to look at. That's why we've got this dye. It's a membrane dye that Mike Watkins has made. So it will associate with the membrane of cells and it will respond to zinc. Now the problem is zinc is very low concentration, so when it's being released you can't really see it as well as you can do calcium. So we're thinking a membrane-bound, localized dye that can detect zinc. Maybe we can see zinc coming out from there. Physiologically, this is the question I get all the time from really difficult, annoying academics. So thank you. Is it physiological? You know, I don't know. I don't really know what zinc is doing in the body. Yes, it's present in the endothelium. It's present in platelets as well. So we've got a hypothesis that says, yes, it could go up at higher concentrations, where it could have an effect. But we've got no evidence for that. We've got no cell model that says we can knock out this and therefore zinc signaling doesn't work. So yeah, it's a good question. We don't know the answer, but you know, zinc is present and it's a hypothesis we're working to. Yeah? Yeah. Not you. Sorry, we've got an expert calcium signaler at the back, and so I'm a bit worried about that. Go on, Claire.
Thanks very much, Nick, for the really interesting talk. I've got a sort of two-part question. So from your model at the end with your last slide, it looked a little bit as if you were hypothesizing a positive feedback loop whereby you get zinc-stimulated zinc release, a little bit like calcium-stimulated calcium release. So is there anything we can learn from the genes that control the well-characterized calcium-positive feedback loop that we might be able to footprint genomically into what could be happening with zinc?
Probably right. I've not really thought about that. I think one of the things I would say is that every single protein that is a calcium-binding protein is also a zinc-binding protein. So the binding sites are better adapted for zinc binding than they are for calcium. But we call them calcium-binding proteins because there's more calcium around. But the change in zinc concentration you get within a cell is around about the concentration at which it would bind to a protein like protein kinase C, for example. So zinc, the change in the concentration, even this small, is enough to affect various types of enzymes. So we think it's, I probably alone in the world think it's quite significant and important and is under study. So yeah, you're probably right. Yes, there are other parallels with calcium signaling that we can look at. And that's indeed what we've done. So we took all the calcium channel inhibitors when we were looking to see how zinc is getting inside the cell to see if we can mimic it that way. But yeah, it's a good question.
I was just wondering if there are any naturally occurring genetic variants where people have certain diseases or disorders that could illuminate this.
Not that I know about, certainly not with zinc signaling, no.
So we do have the calcium expert.
Yeah, we've got Martin at the top there. You're right, Martin.
can you, sorry, it's a bit...
Great talk and amazing photos, by the way. So just to pick up on your comment about zinc binding to sites that calcium would bind to, zinc clearly in your model is doing an awful lot. So what do you think might be the principal responders to zinc for both the acute and prolonged effects that it's clearly having in your system? Is there a calmodulin-like entity, do you think?
I think that is a great question. I'm not saying, I've got to be careful, I'm not saying zinc does everything. I'm not saying, that's the model that we think it can do these sort of things at the concentrations we're expecting. Is there a calmodulin-like protein? Well, most of the zinc is stored in metallothioneins, which are redox-sensitive proteins, which is why I was mentioning some of the reactive oxygen species experiments that Maria was doing. So there's a potential store of zinc in these metallothioneins, which is accessible beyond, following changes to the redox state of the proteins. A calmodulin-like thing, I don't know. I've not really thought about that, but when I've had too many drinks in the pub, I do think calcium's irrelevant to everything. It's all zinc. And I promised Harvey I'd never say that out loud, and I've just done it. It makes me a lot of friends at calcium conferences.
I think we've got one last question from Helen here, and then Nick, you'll be mingling outside, so there'll be lots of other chance to talk then.
Thanks. So the last question isn't about zinc, you'll all be disappointed to hear, but you can answer with zinc if you want to. I've never learned so much about zinc before in my life, Nick.
Nobody else has.
Or heard the word zinc so many times in 1 hour. And that was really interesting. I think becoming a professor is a real time of reflection, as you've just shown us, looking back on your career. What is it that you're most proud of in your career to date?
Oh my God, that's a question I didn't prepare for. Most proud of in my career? I mean, I've got my family life, that's the thing I'm proud of over there. And the other one in Leeds. In my career? God, that's a hard one. I think it's going to be the next thing I do, I think. So I think that initial paper I had, the Watson paper, where I did the initial studies that's saying, we think this is a real thing. A lot of it was based on, previous work, so I can't really say, I came up with this great idea and this is what it was. But I think, sorting out those sort of reactions, sorting out how it was zinc was getting into the platelets, is the most significant thing. I think, if you look at my career, there is a distinct absence of Nobel Prize. That's sort of, it still grates a bit, but, I think that's what I would say is probably that first thing. And obviously things are stemming from that. Like I said, I'm not completely convinced about this at all. A professor in zinc biology and stuff like that, I'm still not convinced that this is real. As the questions I had there, we don't know whether it's happening physiologically or not. And that's stuff I'm still going to worry about. But I can point to another example, many other examples in the platelet field and other fields where people are working on things that are still not proven to be physiologically relevant, and yet they've had tons of research funding. So that's the model I want to follow.
And on that point, thanks a lot.
Thanks very much, guys.
So this is the bit where I say something, really sort of profound and amazing in trying to sort of sum up Nick's talk. I can't do that. I mean, I just thought that was just a fantastic talk, Nick. I mean, a really sort of refreshingly different type of inaugural talk, but really excellent. I mean, I'm very much a non-expert, but the clarity and the elegance with which you presented some really sort of, some clearly very complex research I thought was just fantastic. I thought the honesty in your academic research journey, Christ, we just need to hear more of those sorts of stories.
Yeah, it has been rubbish.
It's not been rubbish. It's not been rubbish. And I'll come on to that in a moment. It's not rubbish, but it is just the honesty of that trajectory, which is just, I think so much needed. And, I think at times we just, we gloss over some of these bumps along the road, which kind of give this unrealistic impression of what research is really all about. But I thought, your description of that was fantastic. I think you are far too modest as an individual and as a researcher. I mean, what you are describing there is that ability to combine a successful research story with successful teaching, the way in which you can describe concepts to a general audience, that comes across, I'm sure, every day in your teaching as well. But also that leadership piece, that ability to, yeah, you've got students at the front nodding, yeah.
I paid them, sorry.
That leadership piece, which again we glossed over quite quickly at the beginning, but that ability to combine all those elements together is very, very difficult to do, and you've done that really successfully throughout your entire career. So I think you're humble, but I definitely don't think you should be as humble as you are. You've got such a huge amount to be very proud of. And then I think the other thing that we haven't really talked about is just the way that you've actually impacted so many people through your leadership roles, but just the general way in which you conduct yourself, both in research and in teaching. Then I know from working with Nick for over a number of years that the number of PhD students, the number of early career researchers that professionally he's impacted in a positive way has just been extraordinary. I mean, he really is. Sort of just I think lift people and encourage people to reach that little bit higher and to keep going at times, because as you all know, it can be tough in academia. So Nick, thank you very much for that. I mean, we are extraordinarily lucky to have you here at ARU. The bit you said right at the end was you haven't got a Nobel Prize yet. Well, you've got 20, 25 years, I'm sure, left. So I think we'll watch that space over the next few years. But Nick, on behalf of all of us, thank you all so very much. And here's just a small token of our appreciation.
Thank you very much.
Thank you very much.
Thanks very much. We do have drinks and nibbles, so please come and have some drinks and nibbles outside, and I'll talk to you all in there.